All right, welcome to the explainer. So think about this. Our genetic blueprint has about 20,000 protein-coding genes. That is a truly massive instruction manual for building a human. Today, our mission is to decode just one single piece of it, a gene called LPAR2, using some incredible data from the Human Protein Atlas. Okay, let's dive right in.
So here's our plan for this decoding mission. We'll start by cracking the code of LPAR2 itself. Then we'll pull its file, check its official ID card. After that, we're going to figure out its job as a cellular doorkeeper. And then, this is where it gets really cool, we'll explore how one gene can create many different versions of a protein. And finally, we'll zoom all the way out to see why mapping our genes is so important in the first place.
Right, so our target is LPAR2. Now I know it might sound like just a random jumble of letters and a number, but for us today, it's our way in. By really understanding this one gene, we get this amazing glimpse into the super vast, incredibly complex world that's buzzing away inside every single one of our cells.
And to do this, we're using the Human Protein Atlas. This thing is a huge international project to map all human proteins. Seriously, think of it like a complete encyclopedia of every single worker in the factory of our cells. We're just going to zoom in on one page, one single entry, to really get what it's telling us.
Okay, first things first, in any investigation, right? We have to identify our subject. So let's pull up the official records for LPR2 and see what its genetic ID card can tell us about who it is. But before we look at that ID, just a quick refresher. What is a gene, really? Well, the easiest way to think about it is like a recipe. It's a specific chunk of your DNA that has the instructions for building one specific protein. And that protein, that's the worker. It's the thing that actually goes off and does a job in the body. So, gene equals recipe, protein equals worker. Simple enough. All right, here it is, LPAR2's official ID. Its official name is, LPAR2. The full name is, a bit of a mouthful, lysophosphatidylate acid receptor 2. Its address? You can find it hanging out on chromosome 19. And its family, or its protein class, is something called G-protein-coupled receptors. Now, hold on to that last one, because that's basically its job description. So we know who LPAR2 is. But now for the really important question, what does it actually do? This is where things start to get pretty cool. You can think of it as a kind of high-tech doorkeeper for our cells. The official data calls it a receptor for lysophosphatidylate acid, or LPA for short. Okay, what does that mean? A receptor is basically a sensor. It's something that receives signals from outside a cell and then passes that message to the inside. An LPA? That's the signal molecule itself. It's like a tiny messenger telling cells what to do. The best way to picture this whole thing is to imagine LPAR2 as a doorbell, and it's built right into the cell's outer wall, or its membrane. It's just sitting there, waiting for a very specific finger to come along and press it. It's not just any old doorbell. It only responds to one specific signal, and we can break that process down into 3 simple steps. Step 1, the signal arrives. An LPA molecule just floats up to the cell. Step 2, ding dong, the doorbell rings. The LPA molecule locks onto LPR2. And boom! Step 3, a message gets sent. LPR2 literally changes its shape and kicks off a whole chain reaction inside the cell, telling it to do something, maybe grow, maybe move, you name it. It's how our bodies communicate at this totally microscopic level.
Okay, so one gene makes one protein, which has one job. Sounds pretty straightforward, right? Yeah, well, biology is almost never that simple. The story here gets way more complex because that single recipe for LPR2, it isn't always read the same way.
Get this. According to the Human Protein Atlas, that single LPAR2 gene can produce 7 different versions of its protein. 7. They're called splice variants. So it's not just one recipe, it's more like a recipe book with 7 different dishes you could make from the same basic ingredients. Here's a great way to think about it. Imagine you have one super detailed blueprint for a family sedan, but the factory, it can choose to ignore certain parts of that plan. It could build the full-featured 4-door sedan, Or it could use that exact same blueprint to build a tiny, stripped-down go-kart with just an engine and a seat. It's the same blueprint, but you get two very, very different machines. And the difference between these versions, it can be absolutely mind-blowing. Just look at this. On the left, we've got the full-featured sedan, a version called LPAR2 201. This thing is a long 348 amino acid protein that weaves back and forth through the cell membrane 7 times to be that perfect doorbell we talked about. But then, look at the right. This is the go-kart, LPR2 205. It's a tiny little fragment, just 17 amino acids. It doesn't cross the membrane at all. It just stays inside the cell. Isn't that wild? It comes from the same gene, but you get these two wildly different proteins that probably have completely different functions. So the really critical point here is that this level of complexity is hiding inside pretty much every single gene. And that brings us to the big picture question. Why? Why do we spend so much time and money and effort mapping all of this stuff?
Well, it matters for a few huge reasons. By mapping genes like LPR2, we start to understand the fundamental rules of how our bodies work. We could identify the roots of diseases. I mean, what happens if your cells start making the go-kart version when they're supposed to be making the sedan? This lets us develop super targeted medicines that could, say, block a faulty LPR2 doorbell. And honestly, it just reveals the absolutely breathtaking complexity that's hidden away inside every one of us.
So the key takeaway from our whole explainer today is really this. When we decode just one gene, LPAR2, we don't find a nice, simple answer. No, instead, we uncover this entire world of function, of incredible complexity and amazing variation. We started with just one name, and we ended with a story about doorkeepers and sedans and go-karts, all of it written into our DNA. And that leaves us with one last thought to chew on. We just barely scratched the surface of one gene, out of around 20,000. So if a single gene like LPR2 holds this much complexity, just imagine, what other secrets, what other stories, and what other functions are still waiting for us to discover in the rest of our genome? Thanks for tuning in.